Symposium series with the Knight campus and the Materials Science Institute. We have two wonderful speakers today. First of all, from the University of Oregon and the Knight campus, Vickie DeRose, who'll be talking about detecting platinum anti-cancer compounds, new methods, and new targets. Vicki received her bachelor's degree from the University of Chicago and a PhD from the University of California, Berkeley, and then went on to do her NIH postdoctoral fellowship at Northwestern University. Following that, she joined the faculty at Texas A&M in 1995 and moved to the University of Oregon as a full professor in 2006 in the Department of Chemistry and Biochemistry. She's an associate member of both the Materials Science Institute and the Institute of Molecular Biology and is currently a Knight campus faculty fellow. Her laboratory's research activities are at the intersection of metallobiochemistry, nucleic acids, and chemical biology. Dan Beecham will be talking from the Thermo Fisher side about probing endosomal and lysosomal dynamics with new generation pH-sensitive probes. Dan, as a senior staff scientist at Thermo Fisher, develops cell-based imaging, high-throughput screening, and biochemical tools for drug discovery and basic science research. He holds an undergraduate degree in chemistry from Willamette University and a PhD from Oregon Health and Sciences University. Came to Eugene, Oregon in 2006 after a postdoctoral fellowship at the University College London and the University of Washington in Seattle, where he studied molecular mechanisms of ion channels and GPCR control of neural excitability. Currently, Dan's work involves neural and cardiac excitability in stem cell models, as well as antibody drug conjugate biology and metabolic states in diseases. So welcome to both of our speakers and thank you. It's a little personal thread for all three of us. We all had some interactions at the University of Chicago. That was my first role out of school, out of my undergrad, and I worked with Deb Nelson there, who Dan's collaborated with during his time here at Thermo Fisher. So I have to catch up on the Hyde Park scene sometime. I do want to take a moment to just make a shameless plug for everybody around some open positions that we have at our Eugene site. These may or may not be of interest to you specifically, but maybe you have some friends or colleagues on campus there. This is a very long list, as you can see. Most of the ones on top have to do with our operations team and manufacturing or quality or demand planning. There are 3 roles down towards the bottom for product manager. And then there are 2 roles at the very bottom that are associated with R&D. One's a staff engineer around software, and the other one is a scientist 3 role within our team, reporting to Michelle Yan. And this would be for like an early postdoc and would be involved in looking at flow and imaging type reagent development. So if you or anyone you know has interest, please feel free to reach out to me directly if you'd like, or send them to jobs.thermofisher.com, or if you go to our website and scroll all the way to the bottom, you can click on careers. That will take you to this screen on the right. And you can either put those job codes in, or you can put Eugene in as a location and it'll show you everything that's available there. So appreciate any engagement that we might have there. Also, I want to point out that by request, we've got a lot of potential discussion that we haven't gotten to in the early seminar series. So today we're going to keep the meeting open for 15 minutes after the hour. So I would encourage everybody that's interested in a roundtable discussion to join Dan and Vicki. I think there are a number of potential areas around both nucleic acid biology and dyes, as well as potentially Frodo, that we'd love to have people engage in and maybe ferret out some possibilities for collaboration. So with that, Vicki, I'm going to pass this over to you. And as we said, it might take a second or two for Vicki to pull up. So we'll just be patient.
Looks like it might be okay. So let's see. Okay, thank you. Everyone see a screen? Looks good? Looks okay. All right. Thank you all for coming. It's a real pleasure. It's exciting to be able to talk about our work here on campus, as it were. This is a view out of our laboratory window. We're in the top floor of Klamath. You can see the night campus here on the right, and it was great to watch it be built. The urban farm, I'm so glad it's still there, and we have the beautiful Coburg Hills. And my laboratory likes rainbows, and so they like to capture rainbows outside of our window. And what I'm going to be telling you about is work that was done by past lab members and current lab members, and it's a real pleasure. But I wanted to start by just giving you a heads up about something. We have a new NSF research training program, newly funded as of last fall, that is really collaborative. It's a really exciting project. It's a cohort-based program centered on sensing and manipulating molecules in complex environments. And we named it the training program in molecular probes and sensors. And Dan, I have to admit, I actually never thought about the molecular probes background here in Eugene. We should have thought about that a long time ago. But but that's, of course, the history of a lot of the exciting science that's done here in Eugene. And what this program is, it's a training program that will involve early research on small molecules that can manipulate or sense or do things in complex environments and move these into areas where they can be impactful. We will incorporate early market-aligned research and innovation training. And I want to point out, this is a collaboration between the Night Campus, the Material Science Institute, University of Oregon faculty, and several departments. We'll have an information session about this this Friday. And for questions, you can see any one of the folks here at the bottom. So, and the applications for this and information should have be going out right around now. So I'll be happy to answer questions about this later. So the project I'm going to tell you about is actually quite the history of it is quite an example of sort of discovery to impact. And this is one of the greatest stories of basic science to worldwide impact that I actually know of. What this is the discovery of platinum anti-cancer compounds, which began in a very basic science question in the 1960s. Rosenberg, Van Camp, and coworkers were interested in the effects of electric fields on bacteria. And so they set up a simple experiment. They put E. coli in solution with platinum electrodes, and there happened to be ammonium salts in the solution. And they applied an electric field. And overnight, what they found was that the E. coli were elongating, but they weren't dividing. So they reasoned that something was going on in this experiment that was blocking cell division. And it was quite a leap, I would say, at that time, to go from, why would you care about something that blocks cell division in bacteria, to the idea that, well, maybe you have something that could block cell division in cancer, which of course we want to do in fast-growing cells and cancer cells. What they did, they found after some beautiful analytical work that what was happening here was that some of the platinum was dissolving and combining with the amine salts to make small molecule platinum amine complexes, which they, within a few years, were injecting into mice. This is clearly a staged photo. There's no gloves on here, but here's Laura Van Camp injecting cisplatin into mice and for monitoring the effects on tumors. So this history starts with very basic research in the 1800s, basic research on bacteria in the 1960s, and moved very quickly into these anti-cancer compounds that were FDA approved. Cisplatin was the initial one, and then carboplatin and oxaliplatin. And worldwide, these are in use in about 30% of cancer patients right now, either in single or combination therapy. They're frontline therapeutics. They're not as targeted as one might like. They're nonspecific, but they enter cells and shrink solid tumors avidly. All of these are platinum compounds. They have 2 amine ligands and 2 exchangeable ligands. Chlorides are oxygen-based ligands. In the cell, these nitrogen ligands stay on, and these sites get exchanged with biomolecules. These compounds, although in major use, have major limitations. They're cytotoxic. They are subject to innate and acquired resistance. They cause kidney damage, hearing loss, and peripheral neuropathy. And these days, although chemotherapy and surgery can actually make quite a bit of headway against certain cancers, these side effects can cause long-lasting influences that might limit quality of life or limit life expectancy itself. So there's still good reasons to study these compounds. And interestingly, there is, for compounds that are in such wide use, no comprehensive description of all of the cellular targets of these compounds. And you can see the challenge. These are small metal-based compounds, and at least one of them has no carbon in it. So these compounds enter cells. They enter sort of the prodrug, and then they get aquated inside of the lower chloride environment or other environment in the cell. The aquated cationic compound binds then to good ligands to platinum, which include sulfur or nitrogen ligands on proteins, and also the nitrogen ligands on nucleic acids. So an example, what you create is a platinum like nitrogen a coordinate bond, which is exchange inert. For all intents and purposes, you could call it a covalent. And if you could, what it means is that if you could actually pull down this platinum, you could take its cargo with it. And when the platinum binds to two nitrogens, this sort of thing is only repaired in a cell through like DNA excision repair systems. There's some molecular recognition, but not a whole lot in these small compounds. So these compounds are known to bind to DNA, and when they bind to DNA, they inhibit transcription and cause cell death. Early on, it was known that they also bind to RNA, and they bind to proteins. Our lab has been interested in RNA as a target. Here's a nice rotating ribosome, for example. There are millions of nucleobases of RNA in cells. They have the same structure as DNA, basically in terms of nitrogen ligands, and they have a lot of different effects in cells. And so we studied platinum uptake in yeast ribosomal RNA, for example, early on. And I like to show this slide this is a complex slide, but it indicates really the complexity of RNA in cells. These are all discoveries that end about 10 years ago with all the different types of regulatory RNAs that there are. Some are in the cytoplasm, some are in the nucleus. We now know that a whole bunch of RNA is transcribed as non-coding RNA, and they have regulatory effects. And so if platinum is binding to RNA as well as DNA, it could very well be inhibiting regulatory pathways that are as yet not understood. And so we were interested in RNA as a target of platinum compounds and ways of identifying where those targets might be. If you're not used to looking at RNA structure, so RNA is transcribed as a single strand, but then it folds up into these complex structures, and you can sort of squint your eyes, they look like a protein. But one of the differences is that each one of these units here carries a negatively charged phosphodiester bond, which means that these RNAs are negatively charged and certainly attract cations and metal sites. And also the structure is really important for function. And one sort of glowing example of that, current example of that, is in the SARS-CoV-2 RNA genome, which is predicted to have quite a bit of structure. These are computational predictions from the DOS lab at Stanford. And one project in our lab is to use platinum compounds as cross-linking agents to help identify these structures both out of cell and in cell. And Dylan Willis is the graduate student on that, and it was funded by an NSF Rapid. So I just wanted to mention that. So in this talk, I'm going to introduce platinum reagents, and I'm going to tell you now about our efforts to detect platinum compounds in cells. And at the end, I'll give you a short story about platinum compounds and specifically effects on the nucleolus. So detecting these compounds in cells. So this is a busy slide, but I want to point out these things. These platinum compounds readily enter, so platinum usually means these platinum compounds. They readily enter cells. When they bind to nucleic acids and other things, they form an adduct that, as I mentioned, is hard to remove. You can use saturated thiourea outside the cell or cyanide. But inside the cell, this blocks polymerases or exonucleases or other processing enzymes. And that's useful also for some bioanalytical methods. And then once they're bound, they're stuck. And so that does help enable pull down methods, which I'll talk a little bit about. So in our first quest to understand where platinum was binding, specifically to complex RNAs, we used some old, I call it old school now, but primer extension. So if you have complex RNAs like the ribosome, if you have a primer that goes about 15 nucleotides in, you can selectively target certain regions of the nucleic acid, and then extend that with the, for example, reverse transcriptase, and the cDNA that's formed will be blocked by platinum. And if you know where this truncation is, you can infer where the platinum is binding. And so Maura Osborne in the lab, who is currently, I'll tell you where the current positions are of these folks. She's with Dicerna Pharmaceuticals working on RNAi therapeutics. Mora did this, mapping various regions around the ribosome of RNA extracted from yeast that had been treated in cells with cisplatin. And to do this, she did old-fashioned sequencing gels. And this enhances, so as a University of Chicago undergraduate, actually I got my start in labs by doing DNA sequencing gels. So these kind of send a shiver of something up my spine. But you see these blocks here. This is where the platinum, this you can infer from here what sequence where the platinum is binding. And it makes sense based on the sequence. So this works, but it's laborious. And so what one would like to do is now use high throughput methods. So Corey Plakos in the lab, who is currently with ArcherDX, which was just acquired by Invitae, I found, he worked out a high throughput method of doing the same kind of an experiment. He took RNA extracted from cisplatin treated yeast. This would be the platinum adduct. You want to find out where this is. And so this procedure uses same thing, but universal primers now. You get truncated cDNAs and then do sequencing and bioinformatics and find out where those truncation sites are. And so this worked. Corey did this with yeast ribosomes. And one thing of interest that he found was that many of the produced sites were around the exit tunnel of the ribosome, which is also where antibiotics tend to bind, which is kind of consistent with this being an accessible, dynamically accessible area of the large ribosome. So this worked, and one could do this with other RNAs in the cell. But in doing this, Corey, recognized this issue. So when you do these high throughput experiments, you're analyzing, millions of reads of cDNAs, and thank goodness for our bioinformatics program, we can analyze these things. But still, you're looking at a lot of reads that you don't need, and what you're looking for is the statistical change that would indicate where platinum might be binding. What you'd rather do would be really to enrich only the platinated fragments. And for that, you'd like to have a pull-down reagent, like an antibody or something like that. But there isn't one that's specific that works that well for all platinated compounds, platinum compounds. And so what we needed was a pull-down method. And so that launched us into a project where we wanted to have platinum compounds that had a small handle that we could use to attach, a small handle that wouldn't interfere too much with where they were binding in cells, but that we could use post-treatment to attach a fluorophore or biotin and then do pull down and enrichment. And we settled at the time on the azide-alkyd cycloaddition reaction as the pull down method, where this is the copper catalyzed reaction, and then we could add an alkyne or an azide to these platinum compounds. And this kind of work has been done by many. I wanted to mention Carolyn Bertozzi, who's going to be here next week, and that's going to be really exciting. So it's been used kind of quite a bit, but before our work, there were no platinum reagents that had azides or alkynes for this use. So embarked on a synthetic project with my colleague Mike Haley, and this is the crew that worked on the initial syntheses, and Mike is still here, but these are the places that everyone else has gone to. And we made a suite of azide or alkyne modified platinum compounds. So this is picazoplatin. Picoplatin was in clinical trials. And so we added an azide to that. And then most of these, however, are chelating compounds. And I want to apologize one time only that my lab does not particularly use IUPAC type names. We kind of make up our own names, and Mike just kind of accepts that. Thank you, Mike. I don't know if he accepts it, but he'll go with it. So these all work. They work in cells and outside of cells. So for example, here's picazoplatin. Here are 2 strands of DNA. One has a GG. Can bind picazoplatin as a faint product band here, but when you do the click reaction with the danzyl alkyne, it lights up in the gel, and that's because of this ligand change inert coordinate bond here, and then the covalent triazole, and we can confirm this by mass spec. So it works outside of cells, and then it also works inside cells. And Rachel Cunningham in the lab was in my sort of nucleic acid aimed lab, but was interested more in proteins, and ironically is now working at Integrated DNA Technologies. But what she did was she was interested in protein targets of these platinum compounds, and so she treated yeast with our azide-containing compounds and did biotin pull down and then mass spec to identify the platinated proteins. And she was particularly interested in endoplasmic reticulum and ER stress. So when we did this experiment, we didn't know how many or what we didn't know how specific these interactions were going to be. And so it was gratifying that in the analysis, there are about 6,000 proteins in the yeast proteome, but only about 152 were statistically pulled down in her experiment. So those would be platinate proteins. And the procedure was validated. She pulled down copper chaperones, which are known to bind platinum in cells. And then in her new results, she found that platinum was also binding to ER stress-related proteins, including this protein chaperone, protein disulfide isomerase. And she also measured stress induction, ER stress induction in yeast cells. And so her contribution was that platinum compounds were causing ER stress, but they were also inhibiting the stress recovery pathways that things like PDI might enhance. So this was a nice contribution to the field. We also could use these compounds to locate where the platinum was binding in cells. This is mammalian cell culture. Can treat the cells with azide-containing platinum compound. This will bind to its targets in the cell. And then we can fix and permeabilize and use copper catalyzed click reaction with an alkyne fluorophore and ask where is the platinum binding inside these cells for fluorescence microscopy. And so we did this with a number of different cells. These are HCC cells. And what we started seeing were, so the green here would be where the platinum fluorophore is located. We see the nucleus of the cells, but we kept seeing these bright spots. And these bright spots are the nucleoli of cells. So we saw this in HCC cells. We saw this in HeLa cells. This is an interesting double click experiment that results in 2 fluorophores per platinum. And so these punctate spots here are the nucleolus of the HeLa cell. And we saw it in MDA cells too. So these bright spots here, these are all the nucleolus. And so at this point, we were finding that these platinum compounds were localizing in other places too, but also in the nucleolus of cells. So that leads me to my last part of what I want to tell you about today. So so I told you a little bit about platinum compounds and our ability to use high throughput methods. And we were localizing platinum to the nucleolus. And interestingly, right around that time, the nucleolus came into interest for other reasons. So the nucleolus of cells is a really interesting subcompartment of the nucleus. It's where ribosomes are made. So the machines that make proteins are synthesized in the nucleolus. And it has a lot of other really important facts in cell biology. And it's been of long interest in cancer biology. So the nucleolus is where ribosomes are synthesized. Cancer cells are growing fast. They need a lot of proteins. They need a lot of ribosomes to make the proteins. And they often have enlarged or changed nucleoli. And it's long been recognized that if one could specifically target the nucleolus, that is another way of targeting fast-growing cells like cancer cells. So there was a great interest that we saw this when this paper appeared. The Bruno and coworkers were investigating cell death pathways incurred by platinum compounds. Mainly cell death pathways, DNA damage response has been thought to be the main way that platinum compounds kill cells. But what they found, interestingly, was that certain compounds were causing cell death, not by DNA damage, but by nucleolar processes or ribosome biogenesis stress. So, so we were initially really interested. We thought, well, we have these clickable compounds. We could use them if they cause nucleolar stress. We could use them to determine what these targets were. But so we set up doing nucleolar stress assays in our lab. A nucleolar stress assay, one of the easiest ones, is to look at the relocalization of this protein NPM1, which in normal cells is in the nucleolus, here in blue. In cisplatin-treated cells, still stays in the nucleolus. But with oxaliplatin, you see it dispersed in the nucleus. And also these nucleoli are rounded. That's a phenotype of nucleolar stress. So we use this to look at a number of different compounds. So the nucleolar stress containing or incurring compound oxaliplatin has the carrier ligand diaminocyclohexane and then the leaving group oxalate, whereas cisplatin is simpler, has these 2 amines and dichloride. So first, this team here started out by testing these Frankenstein compounds, diaminocyclohexane with dichloride leaving groups, and then diamine oxalic acid or dope. And they started asking questions like, does dope cause stress here in Oregon? And they tested both these compounds and a number of different compounds. And so here's a nice survey of them. So here's stress, here are unstressed cells, nucleolar stress, no stress. And looking at this, so these are all single cell analyses for these compounds. When you do this kind of work, it's really helpful to have a population-based assay. And so they developed a way of quantifying these for hundreds of cells, just based on the heterogeneity of staining in the nucleus. And a lower coefficient of variation here means the cells are stressed. So low CV cells are stressed. So now we can look at our compounds on a CV, and these are the other compounds that are stressed. And we see actinomycin D here as a control for nucleolar stress. And then just these 4 compounds, all of them are causing nucleolar stress. So now if we look at what those are, all of those compounds have the ring. So the diaminocyclohexane, diaminocyclopentane, and diaminocyclobenzene here. The ring has to be oriented in a certain way. So if the ring is juxtaposed, it does not cause nucleolar stress. If it's flexible, it does not cause nucleolar stress. And then sadly, our current clickable analogs do not cause nucleolar stress. So we have a limited structure function thing here. The next question was, what step in the nucleolar processes are these compounds inhibiting? And so we looked at initially, the first step of ribosome biogenesis is the transcription of our DNA to our RNA. And Emily Sutton in the lab looked at that using pulse chase experiments, and sure enough, oxaliplatin blocks ribosomal RNA synthesis, whereas cisplatin does not. And then for our suite of compounds, they all blocked ribosome our RNA synthesis, with pentaplatin being slightly less effective. So this is interesting. So inhibiting ribosomal RNA transcription, or polymerase I activity, is so far has only been shown in a few different small molecules. These 2 are in clinical trials. Actinomycin D has long been known to have this effect. You can see there are all these multi-ring compounds. And so it's pretty unusual the way that these platinum compounds are causing this very specific inhibition is of great interest. So we're currently interested in the mechanism of this. One thing that, oops, I forgot to take out some slides. So I combined a bunch of stuff. Yeah, so one thing that we're interested in is the ring size here. I noted that pentaplatin caused a slightly less inhibition of RNA pol I. And Hannah Pig tracked down time dependence of nucleolar stress induction and showed indeed pentaplatin shows less nuclear stress than the other ring-containing compounds. And Matt Iglesias did ICP-MS and showed that this is not because pentaplatin is not taken up in cells. It's definitely taken up in cells. So it's getting into cells, but it's having a less effect. And that's interesting because that suggests that this versus this is changing maybe a lock and key type mechanism or a polymerase bypass mechanism, but it's kind of narrowing down what the possibilities are for the inhibition caused by these compounds. So now we're interested in targets. And so just to kind of imagine what we're looking at here, so nucleic acid, here's the carrier ligand of cisplatin, the diamines on a platinum on a DNA helix. Here's what oxaliplatin or docplatin or anything would leave behind this ring. A slightly smaller ring causes less inhibition, but you can see that this might inhibit some polymerase or something. But there are also proteins that are important in these processes. There's NPM1 itself has a reactive cysteine on it. The platinum could certainly bind to. And a difference between oxaliplatin and cisplatin binding could make quite a bit of an effect. So now we're really interested in locating now the targets of platinum compounds that cause nucleolar stress in the nucleolus. So what we want is a clickable reagent like we have these here, but none of these cause nucleolar stress. We were sad to find out. And our current quest is to figure out the tolerance to substitution, so we might be able to put an azide or alkyne on this ring. And just by way of progress, we know through the work of Haley Smith and then ongoing Andres Guerrero rotation students in the lab, that methyl groups, for example, are tolerated, they still induce nucleolar stress. And so we're inching our way towards being able to modify these for pull-down experiments. Okay, so that's kind of the gist of what I wanted to tell you about today. We're interested in locating these small molecules in cells. They're kind of an unusual quest to look at small metal compounds in cells. And we're interested in tracking down the mechanism by which specific platinum compounds induce nucleolar stress. And this is important both because there are few small molecules, there's very little is known about small molecule interactions in the nucleolus. And also it's possible that there might be some correlation between ability to induce nucleolar stress versus DNA damage and specific types of cancer treatments. So we're going to continue to explore these properties. And these are the students in my lab as of March. They're happily here on Zoom, but I'm hoping that someday we'll be back in person. These are the folks that I mentioned and are at UO collaborators. This work is funded by, we have a lot of support from various entities around campus. I want to call out the bioinformatics program, which has helped in our analyses, and also a couple students have been supported by participating in that program. And then also as a reminder, the NSF, we have this new NRT, which we'll have an information session this Friday. And I want to just leave you with, if during the question session, maybe this is the wish list. So these are the things that we'd be interested in exploring further for our projects, the kinds of things that we like to have. So thank you, and I hope I left enough time for questions, etc. Thanks.
Excellent, Vicki. Thank you. To the audience, anybody, if you have a question right now, we can take it. And again, please think about some of these points here that we can also address during the roundtable. But let's open it up for any questions that have come up right now.
My questions are more suitable for the roundtable, but I also want to make sure that I echo what you mentioned, David, and see particularly if some of our nucleic acid chemists might stick around as well. We have some dyes, I think, that might work in some of Vicki's investigations. But yeah, my stuff is kind of discussion.
Yeah, I have a question. I have to leave my vaccine appointment, but I just asked. I want to be around for the roundtable, so I want to ask right now. So, what is the role of nucleolar stress in other diseases other than platinum, cisplatin addicts? So, is it involved in something other than platinum stress, or just with these compounds?
Yes. So there's a lot that's the nucleolar stress can be induced. The way it's been studied in quite a bit is as a downstream effect, sort of after when cell death starts. Nucleolar stress can be triggered as one of the pathways that happen after that trigger. What's interesting about these compounds is that they seem to trigger nucleolar stress as the primary cause of cell death. Nucleolar stress is otherwise it's a canonical stress response. It gets kicked in under other cell stressors, heat shock, for example. But as I said, mainly downstream of some other primary triggers. And so it's kind of interesting. There are very few ways of really specifically inducing it, like with RNA polymerase I poisons. So and in nature, it doesn't induce, as far as I know, on purpose very often.
Thanks.
I had a
Vicki,
question. I was just wondering, structure activity relationship. As a seven-member ring, like a cycloheptyl ring, was that something that you had tried or at least thought about?
We did not get that large, no. So we stopped at cyclohexane, cyclobenzene. We're bumping out with the methyl groups, but we have not gone larger to cycloheptane. we tried to go smaller, cyclobutane, and we just, that was too strange for the platinum compounds to be made easily. But yeah, we haven't gone larger. It would be interesting to try. Yeah.
And Vicki, when you look at, are you able to just isolate the nucleus or the nucleoli and look at the binding sites of these adducts through other analytical methods like mass spec to understand where they're binding instead of having to pull them down? Or
Yeah, no, that would be a really, so we're working towards that. So right now we have a nucleolus isolation procedure through the hard work of the students. The analysis that, that's a small suborganelle that now if you want to do mass spec or even ICP-MS on it, we're getting down into some challenging analytical methods. But that's a goal, certainly for the experiments. Yeah, kind of an interesting situation. These things that cause nucleolar stress cause some things to leave the nucleolus, but a lot of it is retained. So we just have to be aware of that when we isolate nucleoli from nucleolar stress cells. We're losing some stuff because it's dispersed. So the nucleus versus the nucleolus, I think, is going to be what we're going to be wanting to analyze. But yeah, thank you. That's kind of where we're headed in the bioanalysis world. Absolutely.
okay, well, please, folks, feel free to use the chat function to continue to pop questions in if you have them. We'll go ahead and let Dan move forward, and we'll take some additional questions as we wrap.
Great. Thanks so much, David, for the introduction and the opportunity to join with all of you over at the U. And thanks, Vicki, for a great talk teeing off the importance of structure-activity relationship in the collaboration between chemistry and biology. Of course, the bioinorganic spin brings a new flavor to it that a lot of us don't really get to see too much. I will be speaking today about the biological output of a lot of SAR that we do, with our chemists here in Eugene, and focus on a couple of pathways that utilize the large number of pH gradients within cells. And so I've noted there that I have my protonated form of my PhD there. So by way of the talk, luckily Dave already introduced me, so I can skip a slide or so there and kind of take us through early measurements of the foundations of probing pH gradients within cells, and then talk about the first generation of the new class of pH sensor probes. Really, that happened to be my first project when I came here in 2006. And then I'm going to talk about some much more recent work where we kind of reinvented the whole approach to answer the same question, but with lower backgrounds and improvements to function, and along the way touch on some applications that these tools make possible. So you saw I kind of hate the headshot of me that Thermo has out there. This is what you would see if you saw me pre-COVID, but here in the lab, this is what I look like most of the time. And as Dave mentioned, I have a major in chemistry, a lot of focus on neurobiology and cellular excitability during my grad student and postdoc work. And then got really granular with sodium channel structure function during my postdoc in the Catterall lab before coming here back in 2006 when our campus was pretty well integrated with the Invitrogen Corporation. I also am going to use the opportunity to make a shameless plug, not just for my favorite hobby, astronomy, but also for our local astronomy club, of which I'm the telescope library lending coordinator. And so we actually have a dozen or so scopes that you can just check out and go enjoy the skies with. And I'll be looking forward to our outreach activities opening back up as well with higher vaccination rates and some of our public outreach work that we do on the regular, Sundays at Alton Baker Park, as well as monthly on top of the College Hill Reservoir. So pH gradients abound within cells. These are, of course, as everyone knows, these are log scale numbers here. So 10-fold gradients, between single digit numbers. And They aren't quite as steep as some of the gradients we've exploited for biological probes in the past, like calcium. You know, it's nanomolars inside and millimolars outside, one of the steepest gradients in biology. But pH probes were realized early on as having an important role in understanding cellular, initially metabolic work, and we'll take a harder look at some of these more recent pathways that are of critical importance in the modern world of drug discovery, which is less drug and more antibodies, right? So today's drugs are not drugs, they're antibodies or antibody drug conjugates that target a cell and have to be internalized and imported to a particular environment before they can release a payload. This is the endolysosomal pathway that I referred to in the title of the talk. Along the way, we'll touch on an infectious disease pathway. That this is, you know, specialized cells that have phagosomes, macrophages in particular, will engulf particles and rapidly acidify them. And then also, we won't touch on it here today, but of critical importance in recent work. Let's see here, not so recent anymore. Nobel Prizes, I think 2015 for autophagy and the role of you know, this lytic process within the cells to break down either faulty organelles or just simply degrade unwanted protein. And so I'm going to kind of touch on a handful of factors that will go into the exploration that we have in the pH dyes today. So we know, of course, I mentioned the extracellular pH is neutral. Sometimes it can go lower, though, say in the context of a tumor environment. And there are some tool compounds that are often used either to equilibrate the cytosolic pH with the outside, nigericin, the protonophore. So I'll show some work with that we use to benchmark our dyes with. And then, you know, some known therapeutic compounds, chloroquine, for instance, or baflomycin, or potent toxins like FCCP, which decouple the mitochondrial proton gradient, which is essentially the source of that mitochondria's juice in the separation of charge. Another steep gradient in cells. Mitochondria, of course, have a strong resting negative potential, negative to the cytosol, and that's maintained by the separation of protons. Some of the markers I'll touch on in the biology are to tell us where we are in the cell, the Rab pathway. And there's interest in identifying recycling endosomes and knowing which endosomes are going to be lysosomal and which ones are going to get recycled. And then, of course, phagosomes, of course, as I mentioned, for phagocytosis and macrophage function. And these represent about the most acidic compartment can get within the cell in the lower right there, pH 4.2, 4.5. So let's see here. Fluorescein was first synthesized, I think, 100 years before this measurement in 1871 by Adolf von Baeyer, but it took many decades for people to actually try it out and some derivatives to look inside of cells with this material. And Thomas and researchers in '79 actually made the first the fluorescein derivatives that would then go on to influence a great deal of biological research, and that's the carboxylated form. And he did a series of comparisons of the carboxylated fluorescein and non-carboxylated. And what's kind of cool here is that they would load cells with the dye and then grind them up and actually either do fluorescence cuvette measurements. You notice that the y-axis on the bottom graph there is absorbance. This sort of predates traditional fluorescence measurements, so they just do absorbance measurements. And they discovered and exploited the enhanced pH sensitivity of carboxyfluorescein and saw this nice quenching effect when they loaded cells, and they used it to go on and probe some glycolysis work. Enter Roger Chen as a graduate student a couple of years later, who is frustrated with the loading and retention of carboxyfluorescein. So in what becomes the typical fashion for him, he just invents something new. And he derivatized carboxyfluorescein into a ratiometric form, still reports the pH by the quenching of the fluorescein moiety you see in the xanthine ring up top there. And so, but again, the mechanism being a quench, but you also get the the added benefit of a ratiometric measurement. And he named this molecule something that I didn't have room on the text for here, is just BCECF. But this is one of the first tailored molecules made to go into cells and stay there, become cleaved and trapped inside. And he did some cool measurements, concomitantly measuring cytosolic pH and cytosolic magnesium with what would then go on to become a whole series of ion indicator dyes that we had the great, famous pleasure of working with him over the years. Our first commercial form of one of these dyes was, in fact, this BCECF compound you see in the upper right. And what you can see is an experiment I did on cells that I permeabilized with nigericin, so that I can, pretty quickly equilibrate the inside and outside of the cell. And so I'm just probing the cytosol of the cell, and I can then just change the extracellular pH. And as you can see, the fluorescein moiety, steps down and quenches in response to the acidification. And there's also a significant rundown of that signal. You can see that there's a photo bleaching effect going on all the while. And that's just true of fluorescein in general. It's not a very long-lived fluorophore or stable one. And what we sought to develop was the world's first fluorogenic pH sensor dye. I think we call it out here for the first time, Frodo. And as you might infer from the nomenclature, it is a pH-sensitive rhodamine. So it's not fluorescein based at all. And this AM ester form of it shows, actually this came a little after the first development work with it. It was a later project for me, but this shows the dye getting brighter with acidification. And so this lends itself, people generally like signals that appear instead of disappear with the biology they're measuring. And it makes for also better, if you will, signal fidelity if it doesn't quench. Photo bleach. So you can see we get complete recovery. And you can see that the dye is operating within the range of physiological range, but it definitely does get brighter at near neutral pH. Whoops. And so here are those cells at the neutral and acidified condition. So enter the synthetic work of Alexei Rukavishnikov, our senior staff scientist, one of our senior staff scientists, chemists here. And a couple of years later, I got to collaborate with Timothy Huang in the development of the Frodo red and ultimately green dye as well. And we used the principal metric that we used to identify the dyes in vitro was just simply what's the pKa. We wanted a pKa of we wanted to use the pKa as an apparent metric for the signal onset and have a range that would show you the difference between the inside and the outside of the cell. And we also got to reach out and collaborate with as Dave mentioned, a researcher, Deborah Nelson at the University of Chicago, who had recently done some really great work looking at alveolar macrophages and their compromised activity in cystic fibrosis. And she went on to use some of our prototype dyes and ultimately what became the final commercial form of the dye that generate the video you see right here, as well as the data that you see in our first publication. Actually, my first JBC publication came about as a result of this collaboration where we first fused the dye onto E. coli particles and fed them to cells in her human alveolar macrophage model system. Lucy, her postdoc, carried out the recordings. And so this then goes on to become the next 5 years of my life. Basically, taking this dye platform and then parlaying this addressability into a variety of pathways that we identify, of course, in the literature, but more importantly, talking to scientists at conferences and so forth. And so you can see 4 examples here. Like I mentioned, the phagocytic pathway, this more passive endocytosis pathway by which cells just recycle membranes. I'll show a little bit of that later. I also alluded to ligand or antibody internalization a moment ago. And so the first, first forms of the dye I mentioned were free reactive dye, as well as just labeled E. coli. We'd take, E. coli, freeze dry them and label and purify them. and then you could feed them to cells and see their signal appear, like I showed in the video a moment ago. And then you can, go ahead and you can do cool experiments where you titrate a drug on and prevent the internalization. In this case, it's just cytochalasin. By dismantling the actin cytoskeleton, this signal can't, the bugs can't go in and the signal doesn't appear. Later on, we went on to develop protein ligands and labels. So in this case, the epidermal growth factor, transferrin. We made dextran. I'll show some examples of LDL and the cytosolic AM ester. But I want to take a moment to highlight this experiment on the right. This is going to be a model for us a little bit later on, and something I want to discuss a little in the post forum with regards to Vicki's work. So the signal on the left, of course, is what you get when you feed EGF to HER2 positive cells. And if you just simply pre-treat those cells with an excess of unlabeled EGF, you can then displace the binding and knock the signal down and prove to yourself that you have a specific label. And this winds up being the model that I use for everything else going forward. Some off-label uses of the dye emerged in the meantime. The dextran was used by Wallace Thorson to make a serious upgrade to the turf microscopy measurements that he's made in the retinal ganglion cells of synaptic vesicle fusion. These are a series of imaging recordings that are done concomitantly with a capacitive measurement you see on the right. And then in-house, we had a cool experiment done by Oscar Mandavili showing that the cytosolic acidification we should be seeing, it's control on the left and the protonophore FCCP on the right, which releases mitochondrial protons into the cytosol, also lights this dye up very nicely, giving a nice stress model. LDL is another big one. We had a lot of requests for LDL after the product had been out on the market for a while. And so here again is these are now two controls for specificity. And we did a lot of labeling, trial and error. It's not like we just, throw it, you can't just mix the things together and expect them to work. You have to figure out the ratios and the yields of the, the amount of label per protein and all this and identify pretty narrow specs. But using this assay, we're able to show that you can either chelate the label from the outside with heparin, prevent internalization. This also proves that there's no unwanted dye present. Or again, on the right, you can pre-treat with unlabeled LDL. And so here I take it a little step further. This is kind of fun. So instead of just, the massive overdose of the unlabeled form, I can titrate the unlabeled form onto the cells and then compete for entry and get an apparent KD. And this allowed me to first qualify 3 different batches of the material to show that it's reproducible and manufacturable. And again, it just gives us another layer of confidence that we're making something that is still specific and the site is still saturable. And so then you can, again, go back into the lab and take some, hepatocytes and feed the LDL to them. And so, what you don't see is the 10 micrograms per mil LDL all outside the cell. You just see it after it's acidified in the endosome by way of cholesterol delivery. So here we are now in 2012. We have this family of dyes. They've been around for a while. And actually, I think we're going to fast forward to 2016 or so before we start making some adjustments to it. But one of the things we hear back and can just see for ourselves is that, that this is not there's definitely a non-zero background from these dyes. In fact, if I just take my pencil and I draw in, the approximate position of pH 7.4 or 4.5, there's plenty of signal outside the cells at pH 7.4, owing to the half activation of 6.8. And that gives non-zero reads for in the bottom right here in the case of a live cell E. coli uptake assay. So this is owing to several things. One is the pKa, and the other is just the identity of the dye. These are both fluorescein-derived dyes, both the red and green form. These are bright dyes that get brighter. And what we'd really like is a dim or dark dye that turns on. Some of this also is owing to the mechanism of fluorogenicity. There are several ways you can generate fluorogenicity in molecules, and I don't have time to go into all of those. Probably I don't have permission either. But photoinduced electron transfer is the most sort of canonical way. But again, what you have there is a signal that's partially on that just becomes increasingly dequenched as you withdraw electrons from a fluorophore by the binding of a cationic substrate. There were some solubility issues, and we also wanted to up the multiplex and allow people to work in more color channels. So enter the work of Amy Chen and Yishen Hu, who worked together with me for a couple of, almost 2 years, 18 months or so. And you're only going to see 2 dyes, ACA and ACB. There were some 17 dyes that we synthesized, tested, took all the way through into cells, and you know, either passed or failed, and narrowed it down to these 2. First, our strategy was the first part of our strategy was to move the pKa of the dye to the left, so that the dye would have a later signal onset, providing better specificity in the endosomal pathway. And then we also wanted to redshift the dye and number 1, lower the autofluorescence in the cells. I should have put that as the first priority. Cells have a lot of autofluorescence in green and Tritzy channels, depending on the cell type, but also to, again, allow better multiplex and pulse chase opportunities. The 2 dyes we came up with are, as I show here, A and B, and they have pKa's of 4 and 4.5, thereabouts. And what I show here is that, you know, the degree of label of the dye on my target molecule, in this case, Herceptin, importantly, doesn't affect the signal or the pKa. That's something we also saw. But the way that this went is, you know, Amy synthesized the molecules, purified them, and handed them off to me after some chemical analytical characterization. Yi Zhen would generate enough of this stuff to work with, and then I would take it into the lab and generate the label on a target molecule and try some cell biology out. And so again, I kind of highlighted that the features we're looking for in the lower pKa, and I'm not really showing you the wavelengths here, but this is now in the Psi5 or 647 channel. And so the first thing I wanted to ask of A and B is, do they go to the right place? You know, so when I labeled up the Herceptin, I could go to SKBR3 cells, which express the HER2 receptor, and look at my internalization signal and see, does it co-localize with early endosomes, as indicated here by a fusion with GFP and RAB5? No. Does it show up in late endosomes, as shown by co-localization with RAB7A? And yes. And the same is true of B. So that's good news. And then I can come to my occlusion study, and again, I can pre-treat with nothing, or I can pre-treat with unlabeled form on the right, and titrate on the unlabeled form and look at the overall. I get a few pieces of information here. I get an apparent, you know, activity constant, it's sort of like a KD, and I get an overall signal window over here on the far left in the form of the Vmax. And as you might hypothesize, if you target only the late endosomes or the very late endosomes and the lysosomes, you have fewer objects to count from than you do if you go a little bit earlier in the pathway with a pKa of 4.5 versus 4. So there's one bit of data in favor of the B compound. And a bit more just comes in the form of our vetting this stuff to external scientists. So I'm going to have settled on type B right now, and then we'll return a little bit later. But I wanted to now show how we, you know, then go on to really prove out our value proposition, which is, you know, do we have a lower background compound? So first of all, I can take my B labeled Herceptin and compare it to the legacy, the pH dye, Frodo labeled Herceptin, and here at neutral pH, there's quite a bit of, well, there's measurable signal, and it's essentially flat from the new molecule. That results in a signal ratio that goes from something that's already respectable, 5 and 8-fold, and cell biology is not a trivial number to hit, to something almost absurd, where we have something more like you know, luminescence, where there's a dark signal that turns on. I multiplex it here with transferrin to show that we can multiplex. We make it a cell labeling kit, and of course we can feed it to macrophages. I show Skylar on the right here because he spoke to you last week, and this is another place where chemistry becomes instrumental in helping us understand what we have. Can we package it? Can we manufacture it? Is it stable? What does it look like after we conjugate it? I can't really spend too much time on the others that contributed here, but there's a lot more that goes into it than just the bench science as well. But I'll leave off with this model that I've been kind of showing you off and on a couple different ways. So I mentioned that we vetted, you know, our early data to scientists at GNF, Novartis, Regeneron, Pfizer, and started to ask them, you know, well, okay, so here we've already got a tool set that shows you everything in this pathway: early, middle, late recycling, the canonical photo dyes with their, you know, pKa of 6.8 lights up this whole pathway. And you can see over on the right, I've just drawn in a dotted line, you know, the pH range, and then this kind of represents what you can see, right? So visible signals will come wherever you see a line. And so the A compound, now just very lysosomal in scope, really doesn't turn on to these acidic pHs until you get, you know, around little below 5. And that gives us a very specific signal. And there's great interest in just seeing lysosomes. But important to the antibody drug conjugate folks who need a pH of about 5.5 to deliver their payload by virtue of a pH cleavable linker in their antibody drug conjugate, they love the idea of seeing, as long as they don't see the recycling endosomes in the early endosomes, they wanted to see everything downstream of this. So late endosomes and lysosomes. And that is what led us to compound B. So you can see these candy bar designations of the 3 dyes. This work continued through COVID. We managed to all stay in the lab and keep our teams meeting happening, you know, virtually and through time zones and across many different schedules. Actually managed to launch on time in October of last year. So ultimately, our choice of this compound led to this new family of dyes that really just continue to help enable scientists in studying these pathways. And Vicki, I can't help but wonder, given the fact that your ligands are amino ligands, I wonder what the pKa is and what the acidity of those environments are. So I'll stop there, and I probably have gone over by a couple of minutes now. So I've cut into the discussion time, but I'll stop, and hopefully we can go into some discussion now.
All right, Dan, thanks very much for sharing that journey. It's really, I think, an important dye, certainly for a number of cellular biological implications, and it's a great little journey for your career and the organization. So with that, folks, you know, any specific questions, please raise them. And then, as Dan said, we've got about 10 minutes to have sort of an open discussion here while we keep the meeting open.
All right, well then I will kick it off then by asking Vicki, and then hopefully with some of the chemists still in the group. I thought I'd bring up, I got the chance to work with another, it's actually one of Vicki's colleagues who also works on, inorganic anti-cancer compounds that binds to RNA. And one of the things I proposed and we came up with as a project for her student was to use DNA binding dyes, in this case RNA binding dyes, to probe the extent and affinity of binding of Karen's, this is a ruthenium compound, NAMEA. And we were able to find that NAMEA could displace the binding of some of our RNA dyes, Pico Green and some of the others. And it worked with some and not others. And so that told us that there's some specificity, but it suggested that we could have an in vitro assay that then could ultimately become an in vivo assay in living cells. So if you're able to displace the dye with the compound, then you are starting to get a handle on the binding site. And I was wondering if anyone in chemistry who I know 2 things about dyes. One, that they can be major groove binders and minor groove binders. The rest of the coordination that happens with our nucleic acid dyes in DNA in particular is more mysterious to me. And so I'm kind of hoping we could, offline, Vicki and I have communicated a little about this, but I'd like some thoughts on which dyes she might try, because there would be a great ex vivo assay that then would translate to an imaging assay where you could actually do this in situ and probe in cells to see the displacement and, get some information there. And then the stuff, sorry, I'll just do one more thing, and maybe this is something Vicki and I take offline, but do you know about the clickable pyrimycin analogs that we've generated? Because that might also come into play as a means to double label you've already got the clickable platinum, but you've got this ribosomal interaction complex, and I can't help but wonder if you were to click label every nascent protein at once with the oligomycin tool, what you precipitate, will have, well, number one, it'll be double labeled for your clickable platinum, but you'll also get the protein. You can actually get protein identities then, and that's just the thought that came up when you were describing I didn't realize you had so much going on with the click probes, but that's the other piece I'm hoping that some of the chemists and biologists who have developed our click tools might have some insight. Do either of those ideas have legs with you, Vicki?
Yes, so let's see. The clickable pyrimycin, interestingly, we use that as a in-cell measure of just ribosome inhibition. So in concert with our platinum compounds, but we didn't think too much about the double effect there. So that's really interesting. And I'm really interested, the DNA binding dyes, especially to be used in vivo. There's just it's really hard to get at changes in RNA or DNA structure in vivo. And especially, you can imagine things like if we're interested in whether our platinum compounds are binding to ribosomal DNA in the nucleolus. If there was a way of asking, if there was a dye that bound, ribosomal DNA has a lot of substructure in it. If there was a dye that bound to ribosomal DNA, and then we could show that like oxaliplatin displaced it, whereas cisplatin did not, for example, that would be a really interesting in-cell demonstration that our compounds were binding, and we could look at when they bound and things like that. So that kind of thing could be really interesting. And then the other thing that comes to mind is RNA structure, like SARS-CoV-2 RNA genome. There's a lot of interest in the different substructures that RNA undertakes during the life cycle of the genome of the virus, and a lot of interest in small molecules that might inhibit some of those changes along the way. And there might be some, be more like a fingerprint of dyes that would report on one structure and that would change when the structure changed. I think it's terribly hard to predict what interaction, especially in structured RNAs. So that's more of a investigation, sort of high throughput investigation of what dyes would report on what structures. So those are the kinds of things that come to mind.
Yeah, because what I can think of as a screening approach is which is what I gave to Karen. I just sent her a bunch of dyes, and we narrowed it down pretty quickly with the help of her student to find the RNA specific ones. But the DNA library is considerably larger, DNA binding dye library, and I wonder if we might have, I don't know, maybe you have students with time to spare, but a more focused library that is somewhat informed by the complexes you showed, because we know some of the structures, by their coordination in DNA binding. And so I wonder if any of that looks familiar to the nucleic acid chemists in the audience, like Chris Vonnegut or Wen Jun, or not to put anyone on the spot, but some of you guys have a PhD in these things, and I happen to know those are the dyes you studied.
Yeah, no worries, Dan. And that is where, so we do have a number of dyes that have specificity towards RNA. We don't necessarily market all of them for imaging purposes, and quite a few that are, of course, not necessarily in catalog. But the understanding of the binding motifs, it's something we are actively working on and something I have somewhat narrowed down, but it is certainly a lot more complex than the DNA binding dyes, of course, right? DNA binding dyes, like you mentioned, is either minor groove. Usually major groove is way too large for small molecules to bind, but you also get intercalation. But Vicki, absolutely, if we can feed you some more dyes depending on, I'm not entirely sure what Dan supplied. I wasn't really aware of that collaboration. I'd love to hear some of the details behind that, in all honesty. And see what you guys worked with, and maybe there might be some gaps in what you tested. based on what we've learned more recently and, just from our own personal experiences in the lab as well. Because we have a number of RNA binding dyes, mainly we've worked with in a handful of different, either in vivo or in cell. And so we'd love to work a little bit more and, basically see what came out of that.
I think the initial in vitro screen would be pretty straightforward. You could imagine just a titration curve in a 96 well plate with free DNA.
Mhm.
You then just, pick a probe concentration, somewhere in the midpoint of its, binding in the absence of the drug, and then just look at disruption of any disruption or shift in the binding with the drug.
Mhm.
We just, all outside of cells on a microplate to identify the players, and then maybe migrate it into cells. I will mention that, I didn't send Karen just imaging reagents. I sent stuff, of course, straight from the catalog,
Mhm.
but, some of them were for the qubit. And those are the ones that work. So, I think these guys are agnostic to what, we call the usage or the application. It's the off-label stuff that's usually most interesting in these contexts. So,
Oh, absolutely.
definitely want to pick your brain some more. And I'll share Karen's data on the NAMI-A displacement if you'd like. That was RNA specific. I think from my understanding, it sounds like Vicky's more into the DNA probes, but do you think that we would learn anything with major versus minor groove binders, or are there other motifs in the intercalation that might inform, here's the question, is what happens if you get a positive?
I think that the challenge sometimes is trying to find a way to enforce a certain structure in a 96 well plate. So for example, the DNA, for example, the ribosomal DNA that encodes for ribosomal RNA has, I think it's supposed to be not just coiled, but partially supercoiled and particularly, dense. So it's got certain characteristics. And there are also some substructures called R loops that are formed under a variety of different situations, including in nucleolar processes. That's a combination of RNA and DNA, and actually a specific dye for that, because right now there's an antibody for that, which is kind of a pain to use. So I can imagine, but you have to be able to put the structure in the 96 well plate and then replicate it in the structure you think is happening in the cell. So that gets trickier when it goes beyond single and double stranded DNA, as I'm sure Chris knows for RNA.
Oh yeah, absolutely.
When June mentioned something really interesting here that, and this is something I didn't really highlight, but, one of the things we have to put all of our compounds through the paces with is, does it kill the cells? If this is a live cell reagent, we need to do a whole tox study and then compare to benchmarks and show that, you titrate the dye on and see how long the cells live, or did I interfere with this or that? When June mentions that we probably ought to be considering nucleolar stress as one of the things to look at as we develop some of our live cell DNA dyes, because that's it's interesting because you always see, depending on the dye, you do see the nucleoli sometimes there. And I've always just attributed that to be a density thing, that the copy number of the dye isn't necessarily indexed by the number of molecules that are there. It's the packing of the dye in its compartment. Nucleolus might just make something appear brighter, but it's actually just the same label on
Denser,
more
right?
stuff packed into the small space.
Yeah, Dan, I'll add to that by saying that years ago we did a screen in-house of a whole big list of nucleic acid binding dyes that in cell-free systems were evaluated for their selectivity for RNA over DNA or vice versa. We wanted to have a look at how these actually performed, where they ended up in an actual intact cell, both in live and fixed cells. So we have a pretty good data set if that might inform ongoing discussions around this, because for whatever reason, there are certainly instances where we have, RNA selective dyes that, go much more prominently to the nucleolar region than through cytosolic. Some which, bind mitochondrial DNA better than, than RNA. And then, there are a whole range of selectivities, if you will, just determined empirically in whole cells, laid up against what we had previously evaluated just in a cell-free system. So that could inform some of these conversations, I think, if there's interest. I could definitely bring that data set up. We can look at it. Most of them have been long commercialized dyes, but I think there were a couple of internal molecules as well. So please let me know, Vicki, Dan, and others, if that might be useful for ongoing conversation. Very interesting. And while I'm talking, thank you both, Dan and Vicki. Really interesting talks, both of you.
Great, thank you.
Yeah, let me just follow up real quick. Vicki and Dan, again, thank you. I want to echo Mike's comment there. Great talks. Dan, I don't know if you saw Sam Lester's question about whether or not the pH, any of the Frodo dyes have been used in 3D hydrogels.
Yeah, the short answer is that's one I don't know. I know it's been used in 3D cell cultures, organoids and spheroids and things like this, and those are often embedded in a gel matrix. But the specific use in 3D hydrogels is not a reference I can point to. But it is often used to probe those local microenvironments generated by cancer cells. Part of the Warburg effect is, the increased acidification and as a result of glycolysis in the absence of oxygen in the middle of a tumor. So you have this acidic environment, and people have used the dye to label the cells and form spheroids and probe those conditions, but I don't have a specific answer for the 3D hydrogels. What is the use of a 3D hydrogel? I think of the things that capture the little bits of space debris and things like that, but I'll bet that's not it. What's a 3D hydrogel?
Sam, I think you are still online there. Would you like to expound?
Yes, I'm here. Sorry, I'm currently in the office, but let me see if I can get my point across. With 3D hydrogels, I'm currently trying to build a vascular therapeutic that is three-dimensional, that actually has a morphology that can inosculate with host tissue, and that takes a 3D space. And I wanted to see if I could measure the microenvironment of the pH due to the cellular environment.
Yeah, if there are, we have a few reactive forms of the dye. We have, you can use tradition. If there are free amines, that would be the most straightforward way to label, your hydrogel. If you wanted to say label the hydrogel matrix, and then look at the local effects of the presence of cells or migration and things like this. Free amines with the NHS ester derivatives that we have are possible. If cysteine molecules are present or available sulfides, we have maleimides. And we also most recently developed the clickable forms of the dye for targeted incorporation with either metabolic labeling or in vitro labeling with an alkyne precursor. So if those three moieties are possible to impregnate into your gel, then it's certainly a doable thing. We also just have sort of freely soluble forms that may just intercalate and hang out and show you in the solution phase, what's going on. The 10,000 molecular weight dextran is pretty mobile and reaches into pretty small areas, synaptic vesicles, for instance. In the kiss and run, fusion models that I described by Wallace Thorson, it might also just diffuse into your gel and give a report of the local microenvironment. But you might also, again, it depends on the size of the signal, because that form of the dye is the older form of the dye that is, you can adjust the backgrounds, by the exposures and gain of your camera and all that. And that's how I showed everything I showed you before. But it just kind of depends on the pH range that you expect in your hydrogel, and how it might be influenced by the presence of cells or whatever your experimental manipulation is. But I'm more than happy to direct you to some, or I think one of the things we get to do, it's sort of unspoken here, but I get to tag Mike Janes and have some stuff drop shipped to you if you ever want to try it out. Some of these things are a little pricey to just give a try, and so these kind of off-label exploratory things I think this is another opportunity for us in R&D to help you guys a little with some of the things you'd like to try, but maybe the bar is a little high if it doesn't work.
Really appreciate it. Thank you so much, Dan. And
Yeah.
I'd love to talk more about potential collaborations.
Oh, fantastic. Thanks, Sam.
we are past the 2:15 mark as well now. So Dan, if we could, use you as the point person to follow up with Vicki and the chemists on the nucleic acid binding and follow up with Sam, I think, both opportunities sound like a great possibility for collaboration between our two campuses. And I think with that, we'll just again say thank you, Vicki and Dan, for sharing your work today, and we'll close ourselves up.
Okay, thanks so much.
Thanks, everyone.
Thank you all.